Welcome to Neurosalience, the OHBM podcast. Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bandettini. Here I interview brain scientists and discuss their work, as well as the latest advances and challenges in the entire field of brain mapping. My guest today is Professor Arno Villringer. He is the director of the Department of Neurology at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig. He's also the director of the Department of Cognitive Neurology at Leipzig University Hospital and professor of cognitive neurology at Leipzig University. In addition, he's director of the Mind-Brain Institute Berlin School of Mind and Brain. Arno received his MD in 1984 from Albert Ludwig University in Freiburg and did a short but highly impactful fellowship at the MGH NMR Center in Boston. From 1986 to 93, he was in Munich at the Ludwig Maximilian University Department of Neurology. And from 93 to 2007, he was at Charité University Medicine in Berlin in the Department of Neurology, working up to vice chairman. Finally, in 2007, he took his primary role as director of the Department of Neurology at Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig. Arno is a luminary figure in the field of brain imaging, starting with his pioneering work on developing susceptibility contrast for imaging perfusion while he was doing his fellowship at MGH, and then his pioneering work on developing near infrared spectroscopy, and then using this approach to help validate fMRI contrast, as well as shed some light on it. Since then, he's been working on a wide range of topics, falling into the categories of either methods development or mind-body interactions. He's played a major role in many insightful studies that include those using simultaneous EEG and fMRI and looking at neuromodulation, brain plasticity, subliminal stimulation and processing, as well as resting state fMRI. He's been perfectly positioned over the years and extremely active, not only to add to cutting-edge methods and understanding of the brain, but to carry these over into eventual clinical practice. In this conversation, we cover so many things, and the discussion is far-reaching and goes into depth at sometimes, but is really stimulating. So I hope you enjoy it. All right, Arno Villringer, Professor Arno Villringer, thanks for coming on. It's really wonderful to talk to you, and it's great to see that you're doing well. It's wonderful to have a chance to sort of visit a little bit of your research, since it's been so. I mean, you're a neurologist and you're director of the Max Planck of the neurology department in the Max Planck Institute for Human Cognitive and Brain Sciences. And so I've just been amazed with not only the depth of your research, but also the breadth of it. You've been covering everything from methods to clinical applications and everything in between. So with that, thanks for visiting. Thanks for coming on.
Thank you very much for having me. It's a great pleasure.
So what I've known you for, I think almost 30 years. I've known of your work, and so when you started, you were mostly interested. You got your medical degree, but then you also got into research. So what made you interested in doing research along with medicine?
Yeah, as you said, I studied medicine in Freiburg. I did my medical thesis was in a molecular biology lab, and I realized that doing wet lab work and combining it with the clinic may be too far-fetched. And so I thought about doing something in patients and more on a systems level. And I mean, at the time, NMR was a new technology. So actually my thesis supervisor, I remember, I was like 21 or something when I did it. In Germany, you do the medical thesis in parallel to the medical studies. So I was in the lab for several years.
Wow.
And my thesis supervisor, actually, he was a chemist originally. And he had done NMR spectroscopy. And, he had known these big vertical magnets where you put in a tiny probe. And I remember in our weekly meetings that we had, when he was presenting the newest literature in something like 1982, he was saying, incredible. Now they are putting humans in these magnets. He couldn't believe it. No?
Yeah.
And so, but several years later, it was like the upcoming technology. And actually, I was intrigued by the possibility of doing in vivo biochemistry. So having done molecular biology, biochemistry, I thought, you can measure. I saw these spectroscopy data. I thought, you can look at lactate and maybe neurotransmitters or whatever.
Yeah.
And so I kind of came in the end. I won a stipend by the German Research Foundation.
Yep.
Actually, this was at the time an easy thing. So I sometimes show this to my PhD candidates. Basically, my application for 1 year, nice stipend, well funded, consisted of 1 page, basically saying I'm Arno Villringer and I would like to do research on NMR, and I would like to do it in America.
That's great.
And one has to say, the idea was I already had a job in Munich for a residency, and this guy, my future boss, just started a big program. And so the German funding organization likes to send future co-workers somewhere else to learn something.
Yes.
And so I got this grant proposal or this grant easily.
Yeah.
And then I went to Boston.
Yep.
And actually, I remember that the idea was to do lactate imaging.
Oh, really?
Lactate imaging of the human brain in 1985. So it was quite. and I sometimes make fun of Bruce and Tom Brady, who were Bruce Rosen and Tom Brady, because I met Tom Brady at a conference in Switzerland. And Bruce had just developed this Dixon shift imaging method where you can do them. And it was fantastic. Huh? Yeah. And so the hopes were, this would immediately work. And I remember in 1984 or something, 83, Tom Brady told me, you have to come fast because it will be done very soon. No? So when you come, but there will be new other things. No? Yeah. And then I came to Boston, MGH, and having a great time. And for at the time, I'd say that the difference between doing research or working in general in the US versus Germany was even much bigger than today.
Oh, really?
Oh yeah. It was a big. Okay, I mean, I came as a student basically, so it was but the entire atmosphere, also the collaborative atmosphere, was much different than I actually also later learned it in Germany. So I mean, in Germany later, the radiologist said, "But you are doing neurology, so you cannot do NMR research." So
it's very siloed. Yeah. Okay.
So has also changed now, but so it was really a revelation, and I learned a lot from the attitude, doing things fast, and I also tried to do lactate spectroscopy, at least in animals.
Yeah.
But it was very
hard.
Difficult.
Yeah.
It was hard.
I mean, and we're still struggling with doing spectroscopy well.
Yes,
sensitivity. And it's interesting that you mentioned the Dixon technique, because obviously it works really well for fat because it's far enough away, but isolating these specific chemical shifts that are closer in frequency is Yeah, it's tough.
Yeah, I mean, it was a cool idea. And then, of course, the question is whether it would work for such a low concentration molecule with a small shift.
Yes.
And but I was there to learn also.
Yeah.
And so I was working with different people. And since I had time, I was also running this at the time high field 1.4 Tesla animal scanner.
Yes. Yeah.
And so I did all kind of things. And so I did, for example, was involved in just helping out with perfusion imaging of the liver.
Okay.
And based on agents that Wendy Laufer was developing at the time,
Yes.
new contrast agents. And at some point, I remember I just used the wrong pulse sequence. Instead of a T1 weighted pulse sequence, these were T1 agents.
Yes. Yes.
I used a T2 pulse sequence. And all of a sudden, there was this massive effect.
That's
Signal
interesting.
going
So
down.
discovering that it's a susceptibility contrast is useful as well.
And then I thought, wow, if this is a mechanism, maybe it works also in the only organ where I'm really interested in, namely the brain.
So that's actually, I mean, just to pause there for a second. I mean, this is really, I think it's important to let people know that, and I definitely talk about it here, is that for those first susceptibility experiments in the brain are like the precursors to, what we're doing clinically as far as, well, actually perfusion imaging also looks at, obviously the T1 effect, but also some use the bold effect, or not bold effect, T2 star effect with gadolinium to measure perfusion. And so you were the first, one of the first people, if not the first, doing that. Yeah. So.
Yeah, I mean, there were some people in Germany, Jens Fram were also working on similar things. But they did it later, but published at the same time.
But you were using line scanning. You couldn't even image.
Actually, the story goes like this. I remember I injected a massive dose and I saw it in an image. So I then thought maybe I need a faster temporal resolution. And I mean, I'm a medical person, so I'm not the guy who I mean, Python didn't exist at the time, but who learned programming in during my studies. But at least this kind of pulse sequence programming, I could. Well, we were quite mighty, so it was not a long. And so I did this line scanning, a double slice selective pulse sequence, and so I had which was at the time quite spectacular, a 1 second temporal resolution, and then one could see it.
That's incredible. And I remember studying those papers, after when bold contrast came along, and I remember you had probably one of the first. I mean, there's a person, Fizel, I guess, who was there, who developed the susceptibility model a little bit more. But you had a nice layout of bulk susceptibility effects in your paper, and it was great. And then obviously Bruce, also continued on with that, and
Jack Beliveau, not to forget late Jack.
Yes.
So Jack actually, and this is actually Jack basically took over my project when I went back to Germany, and he in 1985 or 6 was saying, I remember that, something like, I will make the visual cortex lightening up with this. I will make PET out of this.
That's cool.
And in the first fMRI paper, which is not a bold, but which is using bolus,
yes,
he basically did it. 5 years later. So, I mean, of course, there was a lot of work to be done, and Bruce was supervising him. It was a lot of effort, but
that's actually so. Yeah, Jack actually, that's right. In 1991, he published that paper, and that's the first functional MRI paper before bold came along, and then quickly bold came along, and that must have been exciting. I mean, I remember probably when you were doing susceptibility contrast. What's really interesting to me is that the field knew that hemoglobin was paramagnetic when it changed its susceptibility with oxygenation or not, but they always, this is sort of how things happen. This is what makes MRI so exciting, is that, people initially think, oh, well, maybe it's not enough, maybe. And then it took like Ken Kwong to just try it. And so what was and then when you first saw it, then I think you were back in Germany by the time, Bold came along.
Oh, yes. It's also, I remember quite vividly. So I was back in Germany, and I was trained, I was sent by kind of the German funding organization to learn about MRI.
Yeah.
But back in Germany, at least in the first years, I could do little MRI because MRI is radiology, and I was in neurology. So we did some work together, but I remember, for example, that we did some animal work in the clinical MRI scanner, and obviously this radiologist, the head of the department, he had a monitor to supervise every scanner in his office.
Wow.
And at the time we were mostly CT scanner, and I remember that there was an emergency call to my partner, a radiologist. His boss was saying, "What are you doing? Who is this patient?"
There's a neurologist on my scanner.
Yeah, that's strange. That was a little scandal, but yeah. But what I did then, and in retrospect, I think for my career, it was good in a way.
Yeah.
So what I had to do something else. So I did basically do optical work, and the link was the following. At the end of my time in Boston, we had played with perfusion and CO2.
Yeah.
And I remember I did some CO2, 1 or 2 CO2 animals, and the effect was dramatic.
Yeah.
And from this very pilot experiment in Boston, I concluded, actually wrongly, but I concluded that obviously there must be a massive effect on the capillary level. And you look in the literature and it says, yes, there's capillary recruitment, capillaries open and close.
Yeah.
And so back in Germany, in Munich, I did animal experiments in the rat. We were studying microcirculation, but with like laser Doppler flowmetry and things like that.
Yeah.
But then I came across the development of confocal microscopes and later dual. And so we had the idea to use the confocal microscope in vivo. We are actually the first ever to do in vivo confocal microscopy.
Wow.
And so my idea was to confirm the existence of capillary recruitment.
Okay.
And
this was
actually,
1994, about 1994.
Yes. But actually, 1994. And actually, we found the opposite. We found the dilation of capillaries, which was not known.
Yeah.
But the capillaries never completely open and closed. There was a relative recruitment in terms of many more, and a widening of capillaries, and in some capillaries, the corpuscular flow, the flow of hemoglobin of erythrocytes would stop.
Yes.
But there was always perfusion.
Interesting.
And then coming to the Bold signal, we also did this, as I said, laser Doppler. And so with the laser Doppler, we had a good temporal resolution, and we were looking at the blood flow response to viscous stimulation. Okay.
All right.
And so this, of course, and at the time, Jack Belliveau just had developed had this science paper, and I remember at some point, for some reason, I had a phone call with Bruce Rosen.
Okay.
And then I told him proudly, I said, we are doing now studies on the blood flow response with a 1 second resolution in rats. And Bruce was saying, that's interesting. We are doing this now in humans. There is a guy, he said, you might not know him yet. His name is Ken Kwong. And that's how
Great.
I learned about the bolt
Oh,
fMRI.
that's interesting. I didn't know that you were already gone, and then then they started doing this. That's interesting. That's
Yes.
interesting. And so, but I mean, your work though, it's interesting. It worked out really well because, I think, I think collaborating with, I remember Ulrich Darnagel, and just actually, it complemented it perfectly. Sort of, people saw bold effects, they didn't know what to think of it, how to really, if it's what sort of connection with neural activity. And then your group was sort of leading the way in terms of just delving into, with this optical signal that has
Yes.
correlates to bold, that there's, you can modulate it. There's all kinds of interesting things you can do. And it's highly related to neural activity.
Yeah, so it was kind of the physiological basis, working on the physiological basis.
Yeah.
And so I remember then there was this, still for me today, one of the most impressive meetings I ever attended, and you must have been there too. I'm sure you were there. This was in a DC hotel.
Yep.
Yeah, like
Ritz-Carlton.
when some people were fighting each other strongly.
Yes. Yeah, I remember that was like the first sort of like a quick workshop on, but it turned out to be amazing. Yeah,
Yes.
yeah, and that's where Bob Turner wanted to name fMRI MRFN, but that didn't catch on. So I mean, there's all kinds of I mean, in the science.
This I didn't know.
Right?
This detail I didn't know.
And abstract, he's like, let's name it this. But
and I had actually then had some kind poster on like the basis of neurovascular coupling or something. Yeah?
Yes.
So which was kind of a basic neurophysiology.
Yeah.
Actually, at the time, I mean, the concept of neurovascular coupling is known for. I mean, we always quote Sherrington, 1890. Yeah?
Yes.
But strangely, I remember somebody saying, "Why do you call it neurovascular coupling? Do you use a strange word?" And I thought it's logical. And later, for some reason, I wanted to know who has kind of coined the term neurovascular coupling.
Yeah.
And looked up PubMed, and actually it was us. It was
That's great.
So at least in PubMed, I mean, the first neurovascular is on something different. It's like on I think vicinity of vessels to neural structures. But then in a book, did you attend that workshop? When we published a book on optical imaging, basically. But
yes.
In that book, it's in the first one, I think. I had an article, and Uli Dirnagel had an article, and we both have like neurovascular coupling in the name, and that's the first it was ever mentioned.
Oh, that's interesting. Okay.
So it's a nice. I mean, the concept, of course, is relevant, and we didn't invent the concept, but it's nice.
Yeah, and I remember, there were so many debates, and it's interesting too. I mean, certain things have changed a lot in fMRI, certain things haven't, and even though there's been a ton of work, a ton of modeling, a lot of really, really nice experimental work from your group and other groups, it's still a little bit. I mean, we can just delve into that as a little bit before we get into other things of a country. I mean, people don't completely understand, what are the mediators for vasodilation with activation.
Yes.
There could be many, and actually, right, even in the paper that you recently, or not too recently, but you still say, there's this mismatch, this overabundance of oxygenation and flow relative to neural activity. There's different models. There's either for metabolic needs, limited oxygen diffusion, and then finally direct neurogenic sort of vasodilation as well. And it's the answer is, as far as I know, is just it could be all of these in different amounts depending. What do you think is the current state of our understanding as far as that's concerned?
I have to confess that I mean I don't do experiments in this.
Yeah.
So maybe there are some new findings that so first of all, I think in terms of the mediators, I mean, we were working at the time, we were NO and stuff. I think there is a multitude of mediators. And I think for such a fundamental process, it's very important that this is the case. And so I think it's a deliberate of evolution kind of redundancy of mechanisms.
Yes.
I also think, I'm pretty sure that there is kind of a feed-forward mechanism, which basically gives kind of which is fast, which could go via interneurons or also a fast signal to the vessel, which dilates, which is overshooting. And then there are several mediators that are like adenosine, pH, lactate, whatever, that do the fine adjusting. So and I mean, what now does precisely what, and it may also be different between different brain regions, which makes sense.
Yeah. Or different for different neurons.
Are dominant in other different brain regions. Now, the weighing, difficult to say. And also, there's still a controversy about whether it actually starts in the capillary and goes upward.
Yes.
Or whether it starts in the arterial.
Yeah.
To be honest, I was quite convinced by the data that it starts kind of in the capillary and then moves upwards. Makes back to me a lot of sense. Huh?
Yeah.
But I would not say that I'm the ultimate expert now on this.
Yeah, and I don't think actually. I think even the experts, I think they're sort of still, trying to figure it out as well. And there's other things that I think are still unresolved, like, the nonlinearity of bold, for instance. What's it's sort of on my mind now, because I've been seeing so many examples, even with Logothetis' paper. He shows that yes, it's linear within a certain regime of activation, but like you increase intensity, it goes up linearly. But then it seems like it's highly bold overrepresents neural activity at very weak or very brief activations, which could imply there's some, there's less control. It's like on off with the vascularity at the very low levels. So maybe bold, it's a good thing that bold increases the sensitivity, but it's sort of nonlinear. There's all kinds of things like that are still ongoing and we're trying to figure out. And I think, but early on, I remember, I think Uli Darnagel was the first person to maybe coin the term of like watering the whole garden.
That was Bob Turner.
That was Bob Turner. Okay, all right, that was Bob Turner. Yeah.
I'm not sure. Even today, I'm not 100% sure whether I agree. Huh?
Yeah.
So I mean, or maybe it's the question to what extent. No?
Right.
In terms of overshoot, that it's what I just said in the initial response. That is not the same. It's like more quantitatively to the same area.
Yes.
At least in the middle time range, I would think that the neurovascular coupling is quite precise.
Yes.
So there, I would not think that there is too much watering around. I mean, obviously, coupling cannot be much better than the density of capillaries. So that's a limitation. But Bob was clearly thinking of a wider watering. No?
Yeah, and that was sort of potentially to explain this overabundance, because if you don't have that much control, then you'd have overabundance because those neurons aren't active. And so, but I think that I think that theory has sort of been, as you say, countered by other modifications.
Or more refined.
More refined, right? Yeah, and it's fun because that's actually what's cool about doing fMRI methodology, is that you can actually delve into that. You can delve into applications, and all right. So we covered your early work in perfusion. We covered optical imaging, and a little bit, but what was, I remember talking to you in the early 2000s, and you had just started work on looking at EEG and correlating that with optical imaging, or then fMRI bold as well with EEG. And do you want to talk a little bit about that? Because I think actually it's really relevant today as well, and it's becoming more interesting, the story.
Or maybe, if you go chronologically, so we forgot near infrared spectroscopy.
Oh, right, of course. Near infrared. I mentally sort of combined optical, but near infrared spectroscopy. You were the pioneer in that as well.
That was like we did optical imaging with confocal, and then the question was, now is there also an optical analog of fMRI, basically? Huh?
Yes.
And that was then the idea of trying it in humans, no?
Right.
And it quite surprised. I mean, we didn't believe our own results in the beginning, but it quite surprisingly worked. And that was for a long time until I had again access to real fMRI.
And where were you working? You were in Berlin this
Berlin? Was just at a transition. So we did the initial work I did in Munich, and then I got a major grant for this. It's like the equivalent of a starting grant on the German level. And with this, we continued that work in Berlin. And try to validating it. Actually, the question is what validates what was a bit complicated. So we did with Jens Fram's group a simultaneous near fMRI study.
Yes.
And of course, to be a bit, let's say, arrogant, I thought this is a validation of fMRI. I mean, fMRI in terms of that we showed that deoxyhemoglobin goes down.
Yes.
Where there is the fMRI activation, which at some point, of course, I mean, it was clear from all the theories, but you have to show it somehow. Huh?
Yeah.
And in the animal, I think it has been shown.
Yes.
And so that was, and yes, we did a lot of validation studies with PET, with fMRI, and I mean, NIRS is still something we are doing, not very much. It has, I mean, I think it is very often applied in pediatric work. Huh? And Helmut Obrich, who was with me all these years until now, he's doing lots of work also in babies and on language development. Also, other people. There, it really has found, I think, a great application. And and also, I mean, in the field, if you want to do something where you really, so we have a setup now where we combine VR, which you cannot do when you move, you cannot do it in fMRI, VR, EEG, and NIRS. So that's where all these mobile technology comes together.
So you had your first paper, if I'm right, I'm really putting this forward, was in Journal of Cerebral Blood Flow and Metabolism in about '96, I guess, with Kleinschmidt and Fromm, or maybe there were others. Or obviously there were.
That was a combination with fMRI. Yeah?
Right.
And the first NIRS was in '93.
'93 with Neuroscience Letters, I guess. And so right. You've been pioneering, and I remember in your paper you were saying, oh, this is potentially a great technique for bedside sort of assessment, and I know that there are companies that sell like these whole heads.
Oh yes, they are quite successful. Huh?
Yeah.
And I think they're doing great work. Of course, I mean, the question is, you should only do it when it's really better, or when it's suited for a certain thing. And when fMRI is suited better, then you should do fMRI. I mean, you don't. So and in this, let's say, in this concert or in this orchestra of methods, I think it has conquered a certain but very strong niche.
Yes.
Where it is widely applied in the field in newborns and babies.
So how would it be applied in like a newborn? Like, would you, if you're wondering about their oxygenation in their brain, or are you trying to do?
I mean, I'm not in the content of that research, but for example, let's say you want to know when does lateralization of language start in the newborn.
Okay.
Or is there, so you do, so typically you do some tasks where you want to know when does a certain ability appear during development. And then you look for neural correlates. And yes, you can also do fMRI, but that's much more complicated and not so ecologically valid. And also studies, I mean, there's one study where we went on a bike, and you can do Now these are health, and so these kind of things are, of
right.
course.
You could do exercise. You could do all, and it's interesting
Yes,
actually.
exercise.
Yeah.
My former postdoc Patrick Weigert here in Leipzig, he does a lot of work in sports medicine, sports science.
Yeah.
So that's interesting, and I think so. There are a number of applications.
Yeah.
And also, if you, speaking of quantitation, if you wanted quantitative change, I don't think NIRS is very easy because you have this issue of extracerebral contamination. And so there's also an issue of quantitation. Huh?
Yes.
But it's probably, in principle, the signals are, at least the changes, you can quantitate it. Huh?
Yeah. And so, yeah. And actually that's nice because, right, NIRS, you can actually, and also you can get oxidation, you can get blood volume, you can get, and you can look at the relative dynamics of those for whatever reason, if you would want to.
And actually, I think the most promising application hasn't even realized yet.
Okay.
And that is, we have one paper on that. That is fluorescence.
Oh.
So we have one paper, a complicated paper, I have to say, but where we show that in principle, you can measure fluorescence transcranially in human subjects.
That's interesting.
The way we did this was that we injected an agent which is fluorescent and absorbing simultaneously, and there is different arteries so the intracerebral comes first, and then comes the extracerebral. And we could pick up the fluorescent signal from the first appearance, which is fluorescent.
Okay.
So which is intracranial.
Yes.
And in other words, so it is in principle possible. It was a high concentration. The problem is somebody has to develop the dyes.
Yes.
And
yes.
if you you have to develop the dyes, you have to go through FDA clearance in humans and stuff, and in an unknown market situation. And of course, the other limitation is that you don't get into the depth.
Yes.
No?
Yeah.
But
right. Penetration is only about a little bit. Yeah.
But there are many neurological disorders that manifest themselves on the surface of the brain. So I think, in principle, that's a very interesting avenue for future development. No?
Yeah, it seems like it could also be a really, as a neurologist, looking for the potential for stroke. It seems like it would be an easy way to, if you had some sort of calibrated NIRS and some sort, having, assessing likelihood for vascular patency or likelihood for stroke or flow deficits potentially. It might just be better to do MRI with that.
We did a lot of studies. We tried a lot of studies, but in acute stroke, I'd say that MRI just won.
It's much better.
Actually CT.
Oh, really?
So
with contrast agents, and you just see it. Yeah.
It's everywhere. You have a CT more easily available.
Yes.
And 24-hour a day, and I would argue that the MRI information is more accurate, more precise. But if time and accessibility matter, then CT most often has more. Of course, there are a lot of exceptions, and actually, I was in Berlin at the time, and the last thing I did was, I still work actually partly in Berlin, and what I won a grant for an MRI on the stroke unit, which is still there. And with that MRI, a lot of interesting things have been done. So now you don't have the accessibility problem anymore, because it's just adjacent to the stroke unit, and so you can do a lot of studies where you don't have this problem of accessibility.
Yeah.
And that's actually quite interesting. But even at the Charité where this is, it's not 24 hours available.
Yeah.
Because it's like a research funded project, and so you would have to have additional manpower.
Yeah.
And so during the day when people are there, then people go in that scanner.
Okay.
But most patients are still diagnosed with CT.
And actually, I remember seeing a number of people who actually claim that using optical imaging, you can overcome to some degree the penetration effects, and with, you can actually do a little bit more depth specific, or even start doing spatial encoding.
Oh yeah. I mean,
eventually.
it's not. You can do depth resolution.
Yeah.
But just not. I mean, you cannot cover the entire brain.
Yes. Yes.
You can in preterm babies. You can.
Yeah.
But in humans, you get depth. Oh yes, of course you get depth info. I mean, so
you just increase the power and you get more depth, right? Yeah.
Yeah, and I mean there are different methods. I mean the easiest conceptually is a time resolved, time of flight. You measure time of flight,
Yeah.
and then from the duration, you find with modeling you find the deep neurons, no?
Yes.
Deep layers.
Yeah.
And
Okay. Well, all right. Well, yeah, let's go on to so already you were a pioneer in perfusion imaging with susceptibility, and also a pioneer in optical imaging, which grew into a field of itself and helped push BOLD along as well. So now, and this is really relevant here, just to mention it again, doing simultaneous EEG. We've been doing EEG with fMRI. And you had some early work. I mean, in NeuroImage in 2003, you did it with optical imaging, but also in human
fMRI.
And fMRI. So yeah, in 2008 or between that time, right, you did it with fMRI. And it was one of the first work. I think that there was maybe some work, other people doing, I think Robin Goldman was doing some work as well.
Goldman with Cohen.
Yes, with Mark Cohen.
Yes.
Yeah, but I still think there's more to be obtained, and I think your work was interesting because it specifically was looking at this interplay between subcortical areas and visual cortex, and trying to explain what alpha activity was. And so maybe you want to talk briefly about that early work.
Yeah, I mean the motivation for that was really, can we go beyond mere task on off, and can we get a hold on endogenous
like resting state. So resting state was getting big.
Yes, and it was a kind of resting state when people, I mean, the Biswell paper had appeared, but people did not think that much on resting state at the time.
Right.
And the nice thing about alpha, it's spontaneously fluctuating.
Yes.
And little was known on alpha. And but of course, I mean, the one of the theories that it's just a nuisance, it just has no role, whatever, seemed, I thought, always a bit strange.
It seems that would be
the dominant brain. I mean, come on.
Right.
But I mean, that's of course also not a very scientific argument. I have to admit.
The "oh come on" argument, right? No. But no, I think everyone would agree with you though. That is,
and so yes. So and of course it was also not so clear what is kind of the energetic or the blood flow consequence of rhythmic activity.
Yes.
And I mean, to me, at least was not clear, would the signal go up or down? I mean, would BOLD signal increase with more alpha or decrease with more alpha?
Yes.
And so that was, I mean, this Goldman paper just was partially, I mean, it was around, so the data, but they had suggested signal goes down and beat it then a systematic study. And also, you remember that there was always this discussion on activation, deactivation.
Yes.
So does, when we find in BOLD what we call a deactivation, does deoxyhemoglobin go up then?
Yeah.
And this is precisely what we did. So we found this deactivation, and then we looked at the NIRS, and we found in NIRS the opposite. It looks like a hemodynamic response function. It is actually,
Yes.
only that deoxyhemoglobin goes up in this case. So that was for us kind of a, first of all, understanding in this case the deactivation of the occipital cortex with increasing alpha. Also, and we were struggling with accepting this finding, that actually you have a role in that. You don't know, maybe. I tell you.
Okay.
So I tell you that story before.
Okay.
So I think it must have been before that. We had done some study on pharmacological MRI, giving L-DOPA to patients, Parkinson patients.
Okay.
And we had some interesting findings in deep nuclei, in subcortical areas. And I remember we presented a poster, now I can say that. And actually, I think it was Jack Belliveau at that OHBM meeting presented our poster as one of the highlights of the meeting. But after that, I visited you.
Yes.
And you were saying, Arno, let's look at the data carefully. And so, and you basically, I don't know, I must have had the data with me, or maybe you were in Berlin, I don't know. So I met you.
That's when I visited. Yeah, either way. Yeah.
So I met you, and basically you could nicely demonstrate that all we were seeing were movement artifacts. Like,
okay.
so it was very disappointing talking to you, I have to say. So
I'm glad to help.
But of course, the interface of gray and white matter is very dangerous for these kind of effects. No?
Yes.
And so there was never a paper on this, only an abstract. But that's, I mean, a good thing. That's how science works.
Yeah.
So, but then later, I think it was later, we had this finding that beside the decrease of bold signal in the visual cortex, there was an increase in the thalamus.
Yep.
And I remember I said to my colleagues, Petra Ritter, for example, you know her, I said, I'm not sure. Once I was discussing such a thing with Peter Benettini, and it was all movement.
Oh no, I'm the movement guy. No.
Don't show it to Peter. So if I remember correctly, the paper, we discussed this very cautiously.
Yes.
And now, I mean, it just wasn't 100% clear, but I think it's now been confirmed. So actually there was then a PET study, which is not as susceptible to this kind of artifacts. Yeah. Also showing an increase in blood flow or glucose. I forgot.
Yeah. So what do you think's actually the story? What do you think's happening? Obviously doesn't explain what alpha waves are, but there might be some interesting. It seems like alpha waves are more of an indicator. I mean, early on it was mentioned it's this idling brain, but in some sense it could be a very deep story of and I remember seeing a study at one of the cognitive neuroscience computational neuroscience conferences saying alpha waves might be the signal of how the brain updates its world models. And so if everything's okay, then it's the alpha waves are happening. But if it's like things are updated and you're consciously thinking and experiencing, then it's suppressed. So.
Oh, yeah. I mean, we have been working a lot. Alpha is one of the themes that we have been working on since.
Yes.
So first of all, actually, in I think the nicer paper years later by Becker, the question was now that there is this spontaneous modulation of alpha, and when you do visual stimulation, then you also have a spontaneous modulation of the evoked fMRI response.
Yes.
And so in that paper, we asked, does the spontaneous modulation of background alpha, which goes along with alpha, does that explain the variability of the bold response? And the answer is yes.
Huh.
And to a large percentage.
Oh, that's interesting.
It's Becker, Neuroscience in, I forgot, 2010 or something.
Okay.
So that was the next step. And then, of course, Ole Jensen and these people have very nicely shown alpha is an inhibitory, mainly inhibitory rhythm. It seems to, it modulates excitability of the cortex.
Yeah.
And it also is kind of, I'm just preparing a talk on what I call the gray zone of perception. So when you have a perceptual input or sensory input, then at first, how much this is translated into, like in V1 or in S1, into an evoked response.
Yes.
That is dominated by the strength of alpha rhythm.
Okay.
And at this point, it's not yet conscious. So there must be different processes to make it then conscious again, to make it conscious.
Yes.
And the zone in between, I call the gray zone, because there are, for example, it's like in Blindside,
Right.
We have done similar experiments, and alpha plays a role there, where we look at healthy people. We give them a stimulus, now again in the somatosensory domain, and we ask, did you feel it? And they say no. And then we ask, which finger was stimulated? And they say the correct finger. But now we are with healthy volunteers.
That's interesting. So just below conscious perception, but it's somehow getting through in some way.
Yes.
And this is somehow, interesting.
And it seems that alpha regulates the access to this, but then there's an additional threshold to pass to get consciously perceived.
That's interesting. That's interesting.
That's kind of, I mean, we are working on an entire framework, but our data look like that. And I think maybe what I really like, I have now with me a colleague, Vadim Nikolin, who's a really absolutely wonderful EEG expert.
Yeah.
And so I mentioned that alpha determines excitability.
Okay.
And now, how to prove that?
So the more alpha, the more excited. Is it directly proportional? I mean,
inverse.
Right. Okay.
And now, how to prove that? So, and now we did an experiment where we looked at the somatosensory stimulus, and we looked at the evoked response, N20.
Yes.
And we know this first response is strictly excitatory.
Yep.
Now, if you now, and we measured EEG, of course, to achieve that, and we measured alpha. And we took two stimuli. One is a bit stronger than the other one.
Yes.
Now, when you take a stronger stimulus, then N20 is bigger. Makes sense.
Yes.
And then we looked at the effect of alpha. And what is known, alpha is inversely related to the late evoked potential.
Okay.
So, and also you more consciously, but the first N20 becomes with lower alpha, with higher excitability, with presumably higher excitability, smaller.
Yeah, interesting.
And we were thinking about this, and finally, now we think this is the proof that alpha changes excitability.
Okay.
Why? Okay. Now, how do we change excitability? By depolarizing the neuron. And EEG doesn't measure the action potential. It measures the synaptic activity until the change in synaptic, the change in resting potential until you have this action potential.
Okay.
And now, if you are depolarized already because it's more excitable. The distance in terms of microvolt
Yeah.
is smaller until you have the action potential.
Yeah. Yeah.
And that's the reason why N20 becomes smaller.
Interesting. Interesting.
And
And yeah, and evolutionarily, it says, I mean, is this related to your, is this linked to your Science 2003 paper with
yeah,
yeah.
this is a eLife paper in last year,
Okay, this was
2025.
even last year. Yeah.
But the yes, but actually the 2003 paper with Felix Blankenburg basically started this thinking. This was so basically, actually speaking of the gray zone, there we looked at stimuli you never perceive.
Yes. Yeah. Completely subliminal.
And
Yeah.
completely subliminal. And there we find that bolt deactivates.
Yeah.
And also it seems that it leads to a feed-forward inhibition and kind of from the thalamus to the S1.
Yes.
And if you look at the evoked potential, you see it. The evoked potential stops after 50 milliseconds.
Interesting. Interesting.
And also subsequent stimuli are inhibited.
Okay. Yep.
So it seems to be an inhibitory, and actually what we showed later, alpha increases.
So that in some way increases the signal to the noise of the detection of the true signal in some sense, or
Yes, it's a good point, very good point. So the alpha increases, and you could say that's the reason why it's inhibitory.
Yeah.
Because alpha is inhibitory. But most likely the story is a bit more complicated. Most likely it's kind of a gating mechanism. You only want certain stimuli go in, and I'm pretty sure that, and then there's also the top-down, that kind of, in a top-down fashion, the alpha is kind of configured in a way that's, I mean, I don't have the 100% data to prove this, but some indication that kind of, you have a shaping of alpha, of inhibitory activity, and this lets a certain stimulus through and are easier through, and others don't. No?
Yeah.
So that's kind of the. So
yeah, I can't help but think of the experiments that were possible with. I know that in, is it Nijmegen, that people have been looking at sort of alpha as sort of like the signature of like a feedback signal, and then a gamma would be like a feed forward, and then they're trying to do layer fMRI along with this to try to get. I'm sure you're thinking about these things too. You have a
that's going in that direction. I mean, Ole Jensen was the one in Nijmegen who now he's not in Birmingham, but I think he's like the master, and he has done most of the quite exciting studies about the role of alpha, and it's quite complex.
Yeah.
And but we are, I mean, we are getting there. So we're getting closer.
Yeah.
And there, of course, you need multimodal imaging. Yeah. So we do it most of the time separately, EEG and fMRI. Lots of EEG work also.
Yes.
But I think if you would view them together, then you just learn so much more. No?
Yes, I completely agree. And I think that, and it's funny how, it's been one of those things that simultaneous EEG and fMRI has been around for 20 years, but people haven't perfected it. And it'd be great to have some sort of a system that just works as far as that's concerned. I really wanted to, I mean, obviously if you don't mind going a little bit longer, obviously it's really, we're just
like. Sorry, I was talking too much.
Yeah. But no, we have, there's so many interesting things to talk about. But one thing I wanted to mention, and certainly right, the alpha simultaneous EEG story is becoming more interesting as we go along. But one of the things that I think you could speak to, and your papers caught my attention in terms of clinical applications of bold contrast. Many people are trying to go the route of looking for biomarkers for psychiatric disorders, or neurology, looking at network deficit. But I actually think that one of the lowest thresholds of getting to clinical efficacy is some of your work was the early work of looking at mapping the latency of bold resting state to look at flow deficits. It seems like the maps look just as good as with gadolinium.
Yes.
Do you want to speak about that? Because I think it's important
Yes.
as a clinical.
I completely agree. I think that is really close to a clinical application, given also the fact that we learn more and more about side effects of gadolinium.
Yes.
And I mean, the idea was very so simple. Huh? Just look at the delay of the bolt in resting state. Huh?
Yeah, and some people could argue that, oh, bold is complicated. How do you get one delay? But there is a primary, frequency, and
yeah, there's a primary frequency, and yes, of course, there is, and it's a bit complicated. What is your reference signal? No?
Yes.
So is it a vein? But that said, I mean, you're completely right. The maps look very closely to perfusion maps. You can, with contrast agent, you can repeat it as often as you want. And so actually with the MRI system that I was mentioning at the Charité, it's in close collaboration, and actually my wife is running the system,
that's always
with
useful.
others, and so the person who is really pushing this now is Ahmed Khalil, a great radiologist, Charité. And so basically what we have shown is that you can monitor reperfusion, so you can do which allows you also to see the effect of your treatment.
Yes.
And now they are doing it in many stroke patients that they see there. And I think the feasibility has been shown.
Yes.
And now it's really practicability.
Yeah.
So practicability means things like having a program that tells that you have the analysis immediately.
Yes.
So it's not enough to acquire the data and then after 20 patients do the data analysis.
Yes.
But of course, you have to have it immediately. And in stroke, in acute stroke especially, of course, literally time is brain.
Yeah.
But these kind of things have been addressed. They have a tool that gives immediate maps.
Yeah.
And it's not yet. I mean, it's done just as a research study. But I think this research study then, if you have it now in 200 patients, and then you can really see and compare, also in terms of would I have done the correct intervention based on this?
Yeah.
That is very close to a clinical application. I completely agree.
Yeah, and I think the two things, one, right, there's more work to be done in terms of, fully validating it. I think there's some people argue that it might be somewhat related to the vasculature more than, the relative delays might. But I think actually, looking at the maps and looking at your work, of course, there'll be more validation, but it like you said, it's pretty convincing. But like you also said, though, and this is the problem that I think that a lot of people, like that fMRI has in general, is that these processing packages, they need to just work and they need to go fast. And vendors are a little bit less. They don't really, it's hard to disseminate this to thousands of scanners and have a technician just be able to press a button and have it come up. That's sort of a real barrier.
Yeah. The practicabilities, these are really the crucial things in making it clinically useful.
Yeah. But I think actually you're right. Maybe I'll have to have him on the podcast as well, because I think that in itself could be an interesting topic to discuss in detail. Yeah. But I also want to get really quickly, as a neurologist, you've also been studying plasticity, and I've been really intrigued by your work looking at these motor tasks, these balance tasks, and showing within a few days changes in connectivity with learning and with skill learning, but also changes. I was really intrigued by your recent paper and changes in fractional anisotropy as well. Obviously there have been papers on that, but you've sort of followed up on it, and it's been interesting. So do you want to speak to that a little bit as well? Whether it's
the interesting, So first of all, I think what one learns in these studies is that, and it's also a theme of my research work. I mean, we tend to say we have a T1 image. This is structural, and this is functional, and now it's a T1 image.
Yeah.
And it's something that comes out of the scanner, and so it's not so. That's very important when you relate these findings to what happens actually with the underlying physiology. Yeah? So actually, what really was intriguing for me was in this very first study when we did structural measures, VBM. And
Yes.
we also already did resting state. But so after 2 weeks, there were some structural changes in primary motor areas. They were continuing to learn. And after 4 weeks, they were gone.
Wow. Wow.
And so, in a way, if you think about it, I mean, you are a runner. So, I mean, if every time you run, your motor cortex grows, maybe...
It'd be huge, right?
I'm sure that happened, but so it's not possible. So we now know that it's kind of a general principle. So areas have increased activity, also increased volume in VBM, also increased connectivity, and then most of the newly formed synapses, the spines, they are deleted again, and only a few remain.
Yeah.
And actually, in this first study, we found the long-term changes, especially frontally, so in the planning area, prefrontal, and there we found changes in the connectivity to the motor areas.
Okay.
So and then later we looked at very early changes, and actually you find very early changes in, again, T1, and then you wonder what is it after an hour or half an hour.
Yes.
So I'm still not 100% sure what it is. I don't think that. I mean, it's very unlikely that there are new neurons or anything.
Right.
It's rather maybe a combination. So what could happen is that there is, with synaptogenesis, which goes really fast, that there is an inflow of additional cells.
Yeah.
And that could produce this transient effect.
Yeah.
And this disappears. This is after several days, it's gone.
Is it cell swelling potentially? It's like, small amounts of, or
could also.
Yeah.
I don't know. We don't know.
Interesting.
And yes, and then there are also changes in fractional anisotropy, which indicate from these frontal areas that in these white matter regions, when you train for a long time, then you have structural changes which also persist.
And what's interesting, though, I was surprised by the direction, though. I think fractional anisotropy goes down. I would have thought, oh well, there's more connections, so you have more fibers that would go up. But instead, it seems to go down with learning. Learning skill.
To be honest, I'm not sure what it really means in terms of, if you have, let's say, if you have fibers which now are growing, which are orthogonal to the pre-existing ones.
Yeah, that's true.
Like that. So these studies were not with our connectome scanner with ultra high resolution, where you might be able to disentangle that. So I would say that there was a change, and that is strongly indicative of a structural change in fiber tracts. I would be careful in interpreting the direction more or less.
Yeah. I mean, and I think hopefully, we're conveying the idea that there's a lot. I mean, the more you look into these things with better tools, the more the questions come, and then you make progress, and there's real insight being made. And okay. So two more quick things, and I'll have a few open questions. So before the podcast, we were chatting about focus ultrasound, and it's sort of unpublished work of yours. Is there a potential for using that as either a neuromodulator? And this, in general, with neurologists, the various neuromodulation fMRI could be useful for having targeted sites for neuromodulation. What do you think of neuromodulation, and what are you doing with focus ultrasound?
Yeah, so I'm fascinated by focused ultrasound, I have to confess. And so obviously, on the potential side, it's almost endless. No? So in principle, you can get anywhere in the brain. It is known from basic physiological studies, you can excite neurons, you can induce action potentials, you can inhibit neurons depending on the parameters.
Yes.
So the possibilities are almost endless. But of course, a lot of questions, if you do it in vivo, are open. And
yeah,
I mentioned before, so one study we did was with very low intensity ultrasound. We applied it at 2 assumed depths of stimulation,
yes,
which we adjusted via beamforming. And one was just by estimation targeting the thalamus, and the other one just by estimation just below the skull.
yes.
And we did simultaneously EEG, and we found that in the one condition, the EEG signal, the alpha rhythm would go up, and the other condition would go down. And so we had kind of a differential effect, and that is, for me, a strong indication, not published yet, so this is not peer-reviewed data by Peter Banatini. Maybe he finds a flaw. So until you do that, until you find the flaw, I'm hoping for that this is a proof that we have a differential effects depending on the depth of stimulation.
That's cool. Okay.
And that's already cool.
Yeah.
Now, of course, especially if you go to higher intensities, which we probably would need, there's issues of safety.
Yeah.
Reflections everywhere, which could superimpose. And actually, interestingly, it could actually mean that some studies will first be done in patients that could actually benefit from it.
Yeah. Yeah. Okay.
So speaking of the thalamus, so if one could target thalamic areas which are responsible for pain, this could be in elderly people who would not undergo deep brain invasive stimulation. This could be a fascinating possibility.
Interesting. So it's stimulating the thalamus, it's some specific area might suppress might turn off this mechanism for experiencing pain, I guess.
Yes.
Yeah. Okay.
And so on the other hand, we know really little about what precisely happens with our parameters that we're using transcranially with the neuronal areas, no?
Yes.
With the brain areas. So also, if you think about other techniques like transcranial magnetic stimulation, we know more about it. It is not necessarily true that the effects by ultrasound are just the same, only deeper. And as I said, you could elicit action potentials. Maybe, yes, we elicit action potentials, but maybe that's not so useful to elicit action potentials. Or maybe it's very useful. I don't know. I mean, there are other things, of course. If you go to higher intensities, you can selectively open blood-brain barrier.
Yes. Yeah.
That, I think, also has a potential.
Yeah, especially with drugs or something like that. If you want to deliver or something.
Yes, you just deliver the drug specifically there.
Right.
And also there is a very intriguing work by a Alzheimer researcher from Australia. Actually, when I say Australia, now I should mention that this guy, who is now a very well-known Alzheimer researcher in Australia, he was the class comrade of my sister in my little town in my little village in the Black Forest.
Oh my
So it's
gosh.
very funny. So and this guy, now being in Brisbane, this guy applied focused ultrasound in Alzheimer models of rats or mice, I think mice.
Yes.
And the plaques disappeared.
Wow. It just shakes them loose potentially, or sort of increases.
Yeah. I should know more about the mechanisms. Maybe it has to do with blood-brain barrier, but it's a paper, I think, in Science Translational Medicine, so it's high profile. And so, I mean, as you know, we do not even know whether the disappearance of the plaques helps.
Whether
As
that
you
helps
know,
or not, or might be a secondary factor.
Yeah, the aducanumab controversy. It's the same thing, kind of, no?
Yeah.
But that's also a possibility. But actually, I mean, we are primarily targeting neuromodulation, and we are targeting with, so in our just one a quant where we do it, or where the plan is to do it simultaneously with MRI. So the system is can be performed in MRI, and then MRI, and now it's becoming not so clear which MRI sequence will help us to localize the ultrasound.
Okay.
So there is ARFI, which is a kind of localized, ultrasound triggered diffusion imaging.
Okay.
But it may need, because, I mean, ultrasound moves, is a movement basically.
Yeah.
And then it should, and it does, it's been shown, it does lead to diffusion changes.
Oh, cool.
The problem, we don't know yet whether in the low intensities we are using for neuromodulation, this will be enough for showing this effect in this ARFI, acoustic radiation.
That's interesting. That's interesting. It seems like it should be, I mean, right, that'd be interesting to continuously monitor that with diffusion weighting. It seems like you can go up high enough to actually look at
maybe we have to use the very strong magnetic field gradients of our connectome scanner.
Yes.
So that we'll see. And of course, the other technique to potentially localize it is bolt.
Yeah, you're right.
So it's a bit more complicated because we don't know yet what happens to bolt at the site of stimulation. But
it also might just simply, who knows? I mean, it could affect susceptibility in other ways, or just the effect of field gradients around, whatever. If it's vibrating, there might be less dephasing or something like that. It might change the regime. Interesting. I think that's just a matter of time before you find with MRI a way of monitoring this continuously. And it seems like it's huge potential. And as I mentioned before, I'm not sure if I would volunteer for this yet, but I mean, even the mechanism of bold effects with activation, it's sort of like I can picture it like, shaking the neurons until they depolarize, which seems invasive, but it's probably not. And it's probably, that's not the mechanism either. So, I mean,
actually the mechanism is probably there are stretch sensitive ion channels.
Okay.
So actually the same ion channels or similar ion channels that mediate that just, there was a Nobel Prize 1 or 2 years ago for these piezo channels, no?
Yes.
Basically transmit touch, but also another area of our research is the blood pressure regulation in baroreceptors. And so maybe the same receptors are responsible for the ultrasound effect.
Okay, that's interesting. Well, there's so many things, and right. I mean, there's so, and it seems like like you said, it keeps on growing. I was just interviewing somebody looking at real-time neurofeedback as a neuromodulator, sort of the implicit neurofeedback that might be useful as well. I mean, the combination of fMRI with MRI, with all these methods are just beginning. Okay, so there's so much more to talk about, but we've gone so long.
I'm sorry.
No, it's been completely wonderful. It feels like it's only been about 10 minutes. I mean, your work, and you also have covered things like One other paper I just have to mention before we finish up is really, really intriguing work on perception as a function of the phase of the cardiac cycle, which was kind of was interesting. I mean, it seems like you're also looking at these sort of changes in excitability or changes in perception with, how well you can detect sensory input with your cardiac cycle. So maybe briefly mention that, and we'll
So maybe I should, because the things seem sometimes also a bit kind of this.
No,
that.
It's all
So but I just want to. So one theme that we are working on in the last 10 years or so is brain-body interactions. The model system is a somatosensory. All these studies are brain-body, naturally. And the other theme is heart-brain interaction. And actually, this paper is the relationship between the heart effects and the somatosensory perception.
Yes.
And actually, and also here in Leipzig, there's a major project grant for many years on obesity. Also another way of brain-body interaction. So it's a common thread from probably 80% of the work we have been doing in the last 10 years is really looking at brain-body, or sometimes I call it mind-brain-body interactions. And for example, those interactions which relate to the cardiac interaction, or to also to interaction with relate to food ingestions, are rarely conscious. So the idea of unconscious and conscious, and the transition is also a common theme to that. No?
Yeah.
And yes, so this study by Israel is I like it also because it actually shows that most likely via the baroreceptors, the sensory perception is different in systole versus diastole.
Yes.
And now one could, we are discussing still, is this an effect which has any systole versus diastole, any meaning?
Yeah.
Or is it something which just is the kind of it has to be, but the effect of baroreceptors serves some other means.
Yes.
And if the baroreceptors do something, then it just happens that there's also effect during systole and diastole. And we have been discussing controversially among us, with me actually favoring the idea that doesn't mean much during systole and diastole, and it's more relevant for other things. Now I actually think both is true. It's
okay.
So, but maybe to mention also why I think this baroreceptor effect is relevant for other things. So it's blood pressure related, obviously, and in systole you have higher blood pressure than in diastole.
Yes.
And now, if you think of people with high blood pressure, they have very similar effects. So you have less pain sensitivity with higher blood pressure. And memory is changing with higher blood pressure. So and actually well-being. What happens? So in a actually that's under review right now. It's one of my all-time favorite studies. If you have higher blood pressure, you are in a better mood. You have higher well-being ratings.
Yeah. I think I read that in your paper as well. Right. I mean, it's sort of, that's interesting.
So and basically, and this is again the data we have is based on the UK Biobank. This is like not on 500,000 people or something.
Thousands of subjects.
Yes, but so my theory is it relates to this baroreceptor effect, and it also so that's the mechanism. And it could be highly relevant for the development of hypertension, because what it means is that that I feel better when I have higher blood pressure.
Yeah.
And so because now this, I mean, in earlier evolutionary time, when I had to fight the lion, then I better don't feel pain. I better feel good
Right.
to fight the lion.
Yes.
But I didn't fight the lion every day, but maybe every 6 weeks,
Yes.
if at all. But now we have similar effects with the iPhone ringing, or with a deadline, or
Yep.
with some whatever. So we have it 25 times a day.
Yes. Yeah.
And so actually, I think that that this could be a mechanism for certain people to kind of dive into this vicious cycle.
Yep.
And over the long run, it leads to it's one of the
Just chronic
mechanisms
high blood
leading
pressure. Yeah.
to chronic high blood pressure. Huh?
Interesting.
And I mean, this is actually, we're not the first to say that. There were some theories 25 years ago, and they were kind of discarded because they didn't have the means to address this. And I mean, I would not deny that, of course, there are molecular events, there are cellular. I mean, it's not some voodoo
Right.
medicine, but it's, I think, an additional psychological mechanism that leads to this vicious cycle.
Yeah.
And so talking about brain-body interactions, these vicious cycles in obesity and hypertension, and its relationship, how the brain communicates with the body, and that's kind of the theme.
Yeah.
We talked a lot about methods, but that's kind of the theme we are working on in order to understand also these risk factors first of all.
Yeah, and I think that's really interesting because it's been neglected. I think people think of the body and they think that's the body, and then they think of the brain, it's separate. And there's like two separate things, they're not attached. But I think a lot of maybe the answers to mechanisms of body issues or brain issues might be completely linked. I mean, there's obvious, like you were talking about with exercise studies. I mean, with running,
Yes,
for instance,
yes.
I go for a run, and my mood is my state, my brain state is, to me, significantly changed even after running for 30 years. I just, after each run, it's an effect. It's not and I notice it when I don't run. My wife notices it when I don't run.
I can tell you, if I mean, I'm not you're a professional runner, huh? Did you run I just saw on Twitter, or I don't know, on Facebook, you just won.
Yeah.
You are the
Well,
best.
not quite
You are
professional.
best in your age group, in your 30s.
Yeah. Well, yeah. Well, thanks. I do that as a hobby. My track, for the 55 to 59 age group, I won the indoor nationals in the 800.
It's incredible. It's incredible.
Well, thanks. Thank you. It keeps me going.
Congratulations. I mean, so I wanted to say I'm not by far away from that, but I do by cycling in the morning.
Yeah.
At least when I'm in Berlin. In Leipzig, I don't have. But I feel much better the entire day.
Yeah.
When I have half an hour of doing something. So these are dramatic effects. No?
It really is. And so and I'm sure there's many more than that that go both ways. And I think it's great that you're focusing on that, because I think I think that it's difficult though, because, for instance, a physician, it's much easier if someone is depressed, to sort of prescribe medication rather than a regimen of exercise, and it's much easier to take a pill. But the exercise, I mean, it's not only exercise too. It could be other things that are potentially useful, are more procedural, and but yeah, I think that sort of study is what we need more of, and I agree. So yeah. So just to wrap up here really quickly. So you're at the Max Planck. I'm really impressed when I saw all your roles that you played. You're head of neurology. You're the the director of the neurology center. I mean, the Max Planck is interesting in the sense you have the directors at your center. You have Angela Frederici, you have Nick Weisskopf, who actually we interviewed last year, Christian Dollar, and yourself. And so you have your group, do you interact very much, and how does that work out? And it seems like you're still applying for grants, so it's not totally completely funded in that regard. So, and how do you
Yeah, I mean, for
manage the whole
specific
thing?
projects,
Yeah,
like the ultrasound, for example, no? So, or the obesity project, we go for project grants with the university. But of
yeah,
course, yes, I mean, we have close collaboration in the institute. So with Nick, it's very straightforward. It's
yeah,
about MRI, applying techniques, doing it jointly, developing things. And I mean, I'm speaking of developing adjusting pulse sequences for ultrasound detection. I mean, I will not do it.
yeah, yeah, right. Yeah,
And so that's very close. And Angela Federici, I mean, a great language expert. And there the collaboration is also with patients and aphasia patients in her group. So that's kind of a very natural
okay,
interaction. And actually the clinic that I'm directing, which is a university hospital, that basically serves the entire institute, no? So
okay,
for lesion studies.
all right,
And Christian Döller, who is the newest, also now 4 years, there we have joined actually ultrasound. So he wants to stimulate the hippocampus, obviously, and he's a memory expert, and that of course has relationships to our work on patients with Alzheimer, dementia,
yeah,
and actually, as I said, focused ultrasound.
yeah, yeah,
And yeah, that's a very, I mean, we meet regularly. Actually, long COVID actually is also a joint project that we are
okay,
doing
right,
because it
that's
also has
relevant.
implications potentially for memory.
Yeah, okay,
So yes, that's a very fruitful collaboration, and also with the hospital and with the university, which is actually we are on campus. So they are the clinic is about 50 yards, so
oh,
it's like a 2-minute walk.
yes, yes,
So that's very important, I think, because if you're far away, then even if you're in the same town, it's often difficult to.
yeah. And Leipzig's pretty close to Berlin as well. I mean, so you have, and
Yes,
you have
yes,
sort of a indirect, you used to be at Charité and also
yes.
you have some
So
connections there.
I still have strong links to Berlin. So I have a little group there,
Okay,
and I mean my family lives in Berlin, so
okay,
so I stay overnight 3-4 days in Leipzig, and
wow,
but try to be as often as possible, of course, with the family.
yes,
Now my son is studying, but
yeah.
something real, math, not something so so easy applied. I mean, physics is, if there's anything that he would accept as a kind of science, physics would be it.
Well, the brain field needs more physicists and mathematicians to actually,
Yeah,
get a handle on it.
but he's not yet ready for that. He would dismiss that.
So, and you also see patients to some degree at all? Do you still?
I mean, so we have I'm director of a clinic, but I mean, I have a 8-hour contract with that. So I have and basically Helmut Obrich, whom I mentioned before, he
Yes.
is the attending physician. So he's basically doing by far most of the work.
Okay.
And I'm regularly there and supervise it, but I do not do very much clinical work myself. I mean, at the Charité, I was more or less full-time clinician.
Okay.
So,
Yeah. Yeah.
of course, also with a lot of administrative work. No?
Okay. Okay.
And where I also have some emphasis is on graduate schools. So the Berlin School of Mind and Brain was also something where we got great students. I mean, you probably know Daniel Margulis was my
Yes.
first.
Oh my gosh. Yeah.
My first at the Berlin School of Mind and Brain, and Smadar Wadokaro, don't know, Israel, the one we just. She's
Yes.
also Berlin School of Mind and Brain, so we got great students there.
Okay.
And
but your student actually early on, even before that, was Hake Hikrin.
Yeah. Okay. Yeah. Of course.
And obviously he has taken off.
Now head of university.
Yes.
Okay. he was always tall, but now he's yes, we are good friends. So it's really great. So he was vice president in Berlin, and Hamburg is also a great place, and they have also great neuroscientists, Christian Büchel and those people. So he will do very well, I'm pretty sure.
Yeah, and in general, I think that I mean, I've always been impressed with the Max Planck in the sense that they have a system that really works well, I think, and it's set up nicely where they have these centers, their focus of excellence. You have enough stability, enough resources to think deeply and broadly about things. And I think that it's kind of like the NIH. It's a little different.
Yeah, it's great.
Yeah, but it's great. So
I think it's a bit like NIH. So I mean, I don't know the NIH that well, but my impression is it's quite similar. I mean, somewhere.
Yeah, I mean, there's different philosophies. I think, I mean, the NIH has an intramural program, which is like only 10% typically of the funding, and then the other 90% they just give the grants to other universities. It would be nice if there were more intramural sort of. I actually think the type of research being done is different, and I actually think we're all suffering from. The extramural world is suffering from this constant struggle to write grants, and I think it changes the flavor of the science. It's more short term, it's less long term in some sense, and it's more. It's all makes progress, but it seems like it could make more long term progress if there was more stability.
Yeah, I mean, in Germany, at that time, people also in universities had these long-term funding, didn't write grants, and it was a far too stable and not innovative system. No?
Right, you need attention. You need something.
So you need. I mean, so obviously, it was far too little dependent on grants.
Yes.
Far too little dependent on you being active. You got your salary, you got everything, you had your people, and some people didn't do anything. They became a professor and then they stopped doing research. No? Yes. And this of course was, when I came to the US, a dramatic difference. No?
Yes.
But like with many things, there's probably an optimum.
Yeah. There's a balance. I mean, even in Max Planck, I'm sure you get evaluated. I'm sure.
Oh yeah.
And there's, at least in the intramural program, we get every 4 years we get evaluated, and it's not quite the same, but it sort of keeps you honest, and it keeps you sort of looking and on edge and trying to push the field as opposed to just coasting. Yeah.
And yes, it's so. I mean, I can't really judge in the US, but I sense that for some people it's really just running grants. Yeah. And so that might be a bit over the top, no?
Right.
And so it will probably go back.
I can imagine having a hybrid where you get a certain guaranteed startup or something, and that kind of gets your momentum going, so you can then be more successful or something like that. Who knows? I'm not. That's way above my pay scale to sort of think in those things right now, but it'd be fun to try to give ideas. But anyway, so there's so much more we could talk about, but I think we covered a lot of ground, and it's
wonderful. Thank you.
Yeah, it's always wonderful to see you, Arno, and
it was a great pleasure talking to you.
Yeah, well, thank you, thanks, and good luck with everything.
Neurosalience is brought to you by the Organization for Human Brain Mapping. This week's episode was produced by Alfie Wine and Anastasia Brovkin.